SwayamPrabha. Digital India. Educated India.
Dear students, you are welcome in this class. This lecture pertains to the course Agronomy of Oilseed, Fiber and Sugar Crops. The title of this lecture is Insect Pest and Disease Management in Jute and Quality of its Fiber. So, learning objectives of this lecture are to describe the management of insect pests and diseases of jute crop. In this case, we will see different insect pests that infest jute crop and do lot of damage to the crop, and what are the management options for these insect pests. And then we will discuss the various parameters of jute's fiber quality. Let us see some glossary. Apical meristem, specialized zones of growth at tips of plants responsible for extension of shoots and roots. Girdling, particularly referred with respect to the stem. Circumferential removal or injury of bark of a branch, stem or trunk of a woody plant is called as girdling. Gregarious, this is referred with respect to the pest, particularly insects, that live, move or attack crops or other things collectively in a flock or in a group with others' company. So, there are certain insects whose caterpillars in early stages, they live in group and they are smaller in size and they feed gregariously, means in group. Mosaic, this refers to the plant. Presence of some light and dark green patches on leaves of plants, which is mainly caused by viral diseases. Polyphagous, feeds on plants belonging to diverse group, means this refers to a pest, insect pest, which can go or which can damage several plants irrespective of their species. Tax, T-E-X, tax, it is for fiber, a unit of weight used to measure density of yarns. Yarns means kind of threads. It is equal to 1 gram per 1000 meters. Now, let us check the insect pests of jute. Large number of insects affect this crop. More than 30 species of pests, including insects and mite, infest jute from seedling stage to harvest, which causes damage or losses up to 31 to 34%. So, this crop can be affected at any stage by insect pests, starting from germination, seedling up to maturity. Severe infestation of these pests also affects fiber quality, causing loss in tensile strength, formation of knotty fiber, reduction in fiber length, and sometimes there may be changes in color. So, variety of effects are there. They may include reduction in quality as well as reduction in quantity. Let us see major insect and mite pests of jute crop. One by one, their scientific names are given and their order is given in column, last column, column 3. So, hairy caterpillar, scientific name is Spilosoma obliqua and order is Lepidoptera. Jute semilooper, Anomis sabulifera, Lepidoptera. Jute stem weevil, Apion corcori, Coleoptera. Yellow mite, Polyphagotarsonemus, Letus, Acarina. Mealybug, Phenacoccus solenopsis, Hemiptera. This is Hemiptera. And then grey weevil, Mylocers discolor, order Coleoptera, and indigo caterpillar, Spodoptera lutura, Lepidoptera. So, these are actually major insect pests and mites of jute crop. Let us see some minor insect and mite pest. So, stem girdler, Nupsera bicolor, order Coleoptera. Termite, Odontotermus obesus, Isoptera. Red mite, Tetranicus coffee, Acarina. So, these are minor insect and mite pests. And finally, you can see list of new insect pests that have been reported sporadically here and there in different parts of the country. So, leaf miner, Trachis pacifica, Coleoptera, gram caterpillar, Helicoverpa armigera, Lepidoptera, safflower caterpillar, though it comes to the safflower, but it can also infest jute crop. This is safflower caterpillar, Condica capensis, Lepidoptera, green semilooper, Amina octa, Lepidoptera, Mesta hairy caterpillar, Scintillus laus, Lepidoptera, tussock caterpillar, Dasychira mendoza, Lepidoptera, leaf weaver Homona species, Lepidoptera. So, you have seen that there are different orders of insect pests and mite, and they are mostly from Lepidoptera, Coleoptera or some other orders. And see infestation period of pest in relation to, vis-a-vis means in relation to crop growth stage. So, you can see on the bottom x-axis it is 0 to 20 days after sowing, DAS means days after sowing, then 20 to 40, 40 to 60, 60 to 80, 80 to 100 and 100 to 120 days after sowing. So, normally jute crop matures for fiber in 110 or 120 days after sowing. So, during the whole span of this growth period, you can see that during 0 to 20 days after sowing, indigo caterpillar may come, particularly at seedling stage. Then afterwards, you can say during 20 to 40 days after sowing, up to 60 days after sowing. You can see grey weevil. This grey weevil can come from 20 to 40 days up to 40 to 60 days, or you can say 20 to 60 days. And then you can see jute stem weevil and mealybug. This bug can come from 20 to, you can say 80 or 90 days, up to 90 days. And then comes your yellow mite. Yellow mite can come from 40 to, you can see about 90 days. And then jute semilooper, it can come from say 70 to and then 120 days, say up to maturity. And jute hairy caterpillar. So, you can see these jute hairy caterpillar, jute semilooper, yellow mite, they come at a later part of the life of jute plant, and jute stem weevil, mealybug, grey weevil and indigo caterpillar, they come at early growth of the jute plant. Now, we learned what kind of damages are caused by different insect pests, and starting from number 1, and you will also see how these insects can be controlled. So, indigo caterpillar, highly polyphagous, sporadically assumes destructive nature in early sown jute crop, actively confined to seedling or early plant stage, as you have seen in the last figure or picture. Twisting of top leaves and stunted plant growth are the symptoms, and March to April is peak period of infestation, and normally this crop is planted in March and April. So, you can say in early stage its infestation is quite high. Young larvae feed on tender leaves in groups, you can say gregarious in nature. Now, you see caterpillars very voracious feeder, and quite large patches of foliage are quickly stripped, causing heavy defoliation. Defoliation means removing or eating the leaves. Other caterpillars often devour entire lamina. Lamina is major part of the leaf or leaf blade. Typical damage seen in young seedlings which are cut on ground surface by larvae. So, you can see these are larvae are in very large number, and they feed voraciously this leaves of the plant. Management of indigo caterpillar. So, first is hand picking or destruction of egg masses or gregarious larvae in early hours when they are active on the plants. So, collection of their eggs, collection of the larvae is important strategy that can be done manually. Then prior to infestation, on the basis of monitoring, chlorpyrifos 2 ml per liter be sprayed in base of the plant. As you know that chlorpyrifos is an insecticide, rate of application is 2 ml per liter. Early instar larvae can be managed by spraying neem seed kernel extract 5%. Severe infestation, you can spray quinal phos 25 EC at the rate of 1.5 ml, ml means milliliter per liter, or cypermethrin 25 EC 0.5 ml per liter. So you can see here EC means emulsifiable concentrate. Now the next pest is number 2 here is grey weevil. So you can see picture on the right side. These are the weevils, they are grey in color. So adult grey weevils. So damages Tosa jute. Tosa jute is Corchorus oletorius. White jute varieties are immune to its attack. That means it does not attack the white jute, which is Corchorus capsularis. Weevils attack 30 to 50 days old plants, making holes in leaves. In severe cases cause defoliation, means removal of leaves. Mostly unopened apical leaves are damaged. So younger parts, the growing parts are infested by weevil. Damaged holes magnifies with growth of leaf, means they become bigger in size. Grubs, voracious feeder of roots, causing stunted plant growth. So you can see here, number of holes have been made on the leaves. Sometimes they can devour this leaf. You can see a deficit, this infestation on the right side also. So it can, the crop can suffer a lot. Now, excess nitrogenous fertilizer aggravate damage, means excess N should not be applied. Rather, we should go for balanced fertilization and optimum doses of the nutrient should be supplied. A preventive measure in endemic areas, balanced application of fertilizer. Foliar application of azadirachtin 1% at the rate of 3 ml per liter. So you know azadirachtin is obtained from the neem plant and it is used at 3 ml per liter twice at 10 days interval to reduce the infestation of grey weevil. In severe damage, 2 sprays of cypermethrin 25 EC at the rate of 5 ml per liter at 15 days interval may be done. Means 2 sprays of cypermethrin can control this grey weevil. Third important insect is stem weevil. Both male and female adults feed on foliage. Small pinholes seen in lamina due to such feeding. Female makes 1 or more punctures at top nodes where knot is formed. Knot is formed and there are corresponding numbers of grubs inside the plant. Knot here means kind of swellings. Destruction by grub results in withering and drying of crown leaves just above a spot where insect is hidden and leaves below remain unaffected. Now, damage of apical meristem means growing tissues checks vertical growth and adversely affect quality and development of fiber, or plant will stop growing in length. So you can see all these picture 1 on the extreme left, stem weevil grubs. So grubs are kind of caterpillar which do not have legs, they miss the legs. Then adult stem weevil, here you can see adult weevil, stem weevil grub on damaged shoot. So you can see the growth of the shoot will stop here, and this is the grub inside the shoot part. Weevil damage in plants. You can see the last picture where damage has been done by the stem weevil. How it can be managed? So removal and destruction of stubbles and self-sown plants to avoid carry of pest and reduce infestation. After harvest of the crop, all kind of residues should be removed from the field. Sowing of crop, both Tosa and white jute varieties in late April remarkably reduce the incidence. That means it is more common in early sown crop. Balanced application of NPK fertilizer should be adopted. Need-based foliar spray of cypermethrin 25 EC at the rate of 0.5 ml per liter in early hours, because in early hours its weevil is effective. Number 4 insect pest is yellow mite. So both nymph and adults suck the sap from ventral surface of young leaves even before unfolding. That means they also affect the younger leaves. Midrib of leaves curves downward and lamina rolls inward from two sides. Secondary veins wrinkle and give leaf a rough and crumpled look and do not grow to their full size. Leaves turn deep green with coppery brown shades with typical inverted boat-like shape and drop prematurely. So, leaf fall can also happen due to yellow mite. Vertical vegetative growth of crop is arrested. Internodes shortened and significant yield losses can happen. So, you can see adult yellow mite, this is the adult, and you can see yellow mite eggs. This is the mass of the eggs. Yellow mites on jute leaf, you can see this, and these are mites. They suck the cell sap, and then you can see yellow mite damaged jute plant. You can see they are drooping. Leaves are drooping here, and you can see drying of the leaves start here, and twisting. You can see the leaves are twisted abnormally. This management of yellow mite, what can be done to control this pest? In peak summer, crop be irrigated immediately to subside damage and acute stress. So, irrigation can help to reduce the damage. Tosa jute varieties JRO 204, JRO 524 are more tolerant. Means in areas where damage by yellow mite is expected, in those areas the varieties which are tolerant to mite should be grown. Line sown crop harbors less mite population and suffers less. So, therefore, broadcasting of seed should be avoided. Early sown crops suffer more. Instead of March, April, sowing crop escape mite damage. Means in place of March, sowing can be done in April. So, then one can avoid the infestation of yellow mite. Foliar spray of mineral oil at the rate of 3 ml per liter plus neem oil 3 ml per liter twice at 35 and 50 days after sowing can control this insect. Otherwise, one can go for 2 spray of Spiromacifan 240 SC at the rate of 0.8 ml. See, the quantity is very small, 0.8 ml per liter of water at 36 and 46 days after sowing. Next insect is mealybug. Apical meristem is most susceptible part. Distorted and bushy shoots, crinkled and or twisted bunchy leaves, plants stunted and dry in severe cases. Damage mostly caused by immature stages of mealybug which suck the sap. Vertical growth of plant is arrested with shortened internodes giving bushy appearance or kind of rosetting appearance. Now, you can see the mealybug how it looks. Male mealybug, you can see this one, and then female mealybug. It is different, they are different, and then mealybug on jute plant. You can see they are on the terminal part, the growing part here, and they are sucking the sap, and then the leaves and other parts may start drying. You can see mealybug infested jute plants. So, this is how they are in large number and they get accumulated in a series. Now, preventive seed treatment with thiamethoxam 70 Ws at the rate of 5 gram per kg seed. Systemic insecticides are more effective against mealybug crawlers. Foliar spray of profenophos 50 EC at the rate of 2 ml per liter. Now, next insect is semi-looper. It is called semi-looper because when he moves, when he walks from one place to another place, it makes a loop. You can see in picture on the right side that it is making a loop. This is loop, therefore, it is called as semi-looper. The loop is half, not complete. So, damage starts from unopened leaves, which represent most susceptible portion, or rather you can say most youngest portion of the plant. So, about 95% damage restricted to 9 fully opened leaves, or you can say first fully opened leaves from the top. Newly hatched larvae start feeding leaves voraciously. As leaves grow bigger, cuts become evident and edges of tender leaves eaten and serrated. Severe infestation, what happens? Growing points eaten and destroyed. Stems totally defoliated. Defoliated means the leaves are removed and induce profuse branching. Internodes are shortened. You can see symptoms on the right side. What can be done to prevent or to control the semi-looper? Balanced use of fertilizer is important, and plow infested field after harvest to kill the pupae. And in other case, if possible, the crop rotation should be followed, and deep summer plowing can also help. Spray Bacillus thuringiensis formulation at the rate of 1 kg per hectare. So Bacillus thuringiensis is a kind of bioagent. It is bacteria. It can control this particular pest. EIL, economic injury level for semi-looper, is 10% plant damage at 55 days after sowing. So if it crosses this limit, then one can opt to apply insecticides. So whenever damage reaches 15%, spray fenvalerate 20 EC at the rate of 1 ml, 1 milliliter per liter of water, or cypermethrin 25 EC at the rate of 0.5 ml per liter of water. Approximately 500 to 600 liter solution may be enough for 1 hectare. Insecticide sprays need to be target the apical portion, because most of the larvae are there in the apical portion or top part of the plant. Jute hairy caterpillar. Young larvae feed gregariously, scrape chlorophyll mostly on under surface of leaves. Later disperse to entire field, defoliate older leaves and may completely skeletonize plant. Damaged leaves give an appearance of net or web, and under severe condition, complete defoliation can happen. Causes damage during June and continue till mid-September, coinciding with 60 to 100 days old crop. And what can be done to manage it? When caterpillars disperse, spray lambda cyhalothrin 5 EC at the rate of 1 ml per liter, or indoxacarb 14.5 SC 1 ml per liter. SC means the soluble concentrate and EC means emulsifiable concentrate. So their rates are same, 1 ml each. You can use either of these. And you can see jute hairy caterpillar larva. It is easy to recognize or identify, but difficult at gregarious hairy caterpillar on jute. So you can see they are very large in number. They are smaller in size and they can defoliate the plant completely. You can see picture on the right side how big damage has been done to the crop. Hairy caterpillar damaged jute field. Next is stem girdler. So damage mostly observed in young plants. Damage caused both in fiber and seed crop. Adult beetle girdles stem at 2 places before it starts oviposition. Oviposition means laying down the eggs. 3 punctures or slits or holes made within 2 girdles and middle slit used to lay single egg. Because of girdling, nutritional transfer arrested and stem above lower girdle die and dry up. Branching takes place below the lower girdle of such stem, means there will not be further growth above the girdling part. After hatching, larvae feeds on pith tissues and move downwards along the central hollow. So you can see typical symptom of stem girdler damage here. So at the top, it has girdled the stem and stem is now drooping and the leaves are drying. And similarly, in other case, you can see the girdling was done here and then the stem is curved and it is hanging or drooping down, and these leaves will dry. So stem girdler beetle damaged jute plant on the right side. Now next is red mite, common on white jute, lives under web and sucks the sap. from lower surface of leaves. Incidence is sporadic, but occasionally it causes serious damage to the crop. Affected leaves leathery, turn yellow and drop off prematurely, means the leaves can fall. So you can see on the right side, the sucking of sap has been done on the leaves, and then these leaves have turned yellow, and after some time they can dry and they can fall from the plant. Next is safflower caterpillar. It occasionally feeds on tosa jute. Larvae in early stages makes holes in leaves and feed voraciously as they grow. You can see, you can identify this insect, safflower caterpillar, on jute leaf. This one, it is greenish in color. On the right side, you can also see this caterpillar. Now you have seen many of the insect pests that damage this crop and how best you can manage. Let us discuss some diseases caused by certain agents like bacteria, fungi, viruses and nematodes. So among all diseases of jute, stem rot, Macrophomina fasciolina is economically the most important. Means this disease, stem rot, causes the most damage in India. Although commonly known as stem rot, but any part of plant may be infected by this pathogen. Means it is known as stem rot, but it can also affect the roots, it can affect the branches, it can affect the leaves also. So many parts of the plant can be affected by stem rot, but largely it is named as stem rot. So it produces various symptoms like damping off. Damping off is done in younger seedling. Seedling blight, leaf blight at seedling stage, stem rot, collar rot, stem break, root rot at adult plant stage, and brown spots on pods. So you can see all these kind of diseases are covered under one heading, which is called as stem rot, because fungus is same. It can go for damping off, it can go to the roots, it can go to the pods, it can go to the branches, to any other part. Other important diseases are anthracnose, black band, soft rot, jute mosaic and root knot nematode. In Hooghly, as you know, Hooghly is in West Bengal, Hooghly wilt, Ralstonia solanacearum serum is the actual pathogen, whereas stem rot fungus and root rot nematode help in facilitating the entry of original pathogen. Means Hooghly wilt is caused by Ralstonia solanacearum, but it is facilitated, it is helped by two other organisms. One is your root knot nematode and other is stem rot fungus. So see, it is complex of all these three. Now diseases of Corchorus olitorius and capsularis. So you can see different causal organisms here. So stem rot fungus is Macrophomina fasciolina. Number 2 is Hooghly wilt. It is by bacteria and the name is Pseudomonas solanacearum, but it is facilitated by Macrophomina fasciolina and Melodogani incognita. This Melodogani incognita is nematode, and both may facilitate its entry of this bacteria into the plant system by doing some injury, and from those injured part bacteria can easily enter the plant system. Now anthracnose is fungal disease caused by Colletotrichum corcorum or Colletotrichum gliosporides. Then black band, fungal disease caused by Botryoidiplodia, theobromine. Then soft rot, fungus disease, Sclerotium rolfsi. Tip blight, fungal disease, Curvularia subulata. Stem gall, fungal disease, Physoderma corcori. Powdery mildew, Odium species. So you see, most of the diseases in jute are caused by fungus, but of course one or two bacterial diseases are there. Similarly, fungal diseases are also there. So you can see Cercospora corci, it is causing sooty mold of pods. Cercospora corcori, and then Corynespora cassicola and Alternaria species. So, these 3 species can cause sooty mold of pots. Then dieback fungus disease, Diplodia corcori, root gall nematode. So, Melardogani incognita and Melardogani javanica, these are 2 species which can cause root gall nematode disease. Then jute mosaic viral disease, and this virus is transmitted by whitefly. So, we will see the details of these diseases in jute crop and what are important control measures for them. Then also certain viral diseases, more viral diseases like jute chlorosis. It is by virus, a member of Tobra virus genus, and then yellow vein disease, a bipartite bigomo virus, and vector is whitefly. The scientific name of whitefly is Memecia tabesi. So, let us start with the most important and significant disease of jute, that is stem rot. Although commonly called stem rot, but any part of plant may be attacked by the pathogen or attacked by the fungus. For example, in damping of newly emerged seedlings rot above and below the soils, means mostly on the crown region where stem and root part meet each other, or you can say where shoot and root part meet each other, crown part, there fungus attacks and rotting happens and seedling can dry. In seedling blight, cotyledons become brown to black and later young seedlings can also die. In leaf blight, during high humidity and rainfall, leaf becomes brown. So, all are being caused by the same fungus. Now, you can see pictures and symptoms at seedling blight. The first one, blighting means just drying of seedlings, they can dry, and damping off, you can see certain seedlings are dead, and leaf blight. These are the symptoms of leaf blight, and then it can cover the entire leaf also. As a result of stem rot, in June to August, leaves become brown in color due to prevailing high humidity. Dark brown lesion on green stem may extend vertically or horizontally. Plants wilt, wilt means dry up, and leaves droop or stems break in high speed winds. So, you can see on the right side this blackening of the stem part, and there can be breakage of the stem part. Infected plant dries up and slowly becomes dark brown. You can see dark brown or blackish. Collar rot is another symptom of stem rot. Brown rot in collar region. Root rot, plants wilt, defoliate and become brown to black later and stand as naked stem and finally die. So, you can see the stem rot here, the all rotting of the stem, and then every part of the plant will dry up. You can see more close symptoms of stem rot. So, here the translocation of food material, water and nutrients will be adversely affected. And you can see the symptoms on the right side also. These are all symptoms of stem rot in jute. You can see more pictures, drooping of leaves. Drooping means just they are hanging from the branches or from the stem, and this is the situation. This is severe infestation of stem rot. So, you can see the plants are completely defoliated. There are no foliage or leaves on the plant. So, roots are rotten and brown and rootlets absent. So, you can see this stem rot fungus can also affect the root part, and once the root is rotten or dried, then the whole plant will dry. If branch roots are attacked, plants may grow and remain alive with reduced vigor, showing rusty brown branch roots. If the tap root or main root is infected, the main root is this one, the main root is infected and damaged, plant generally dies. What is the management of stem rot? It is soil-borne, seed-borne as well as air-borne disease. Crop rotation can be adopted, particularly taking non-host crop in the rotation like rice, wheat, mustard may be rotated. Seed treatment with carbendazim 50 Wp at the rate of 0.1% active ingredient per kg seed. Spray copper oxychloride 50 WP at the rate of 5 to 7 gram per liter of water. Or if copper oxychloride is not available, then tabuconazole or hexaconazole can also be sprayed at the rate of 1.5 to 2 gram per liter of water may be sprayed. So many options are there, including seed treatment and foliar application by certain fungicides. Anthracnose, another important disease of jute, sunken spots of various colors on leaves, stems, fruits or flowers. Irregular spots on stem, enlarge, coalesce means they merge into each other. Deep necrosis means death. Cracks and exposed fiber slowly turn to brownish depressed spots. So this is how you can see the symptoms of anthracnose on this stem part. Leads to wilting, withering and ultimately dying of infected plant tissues. Depressed spots observed on pods also. Infected seeds lighter in color, shrunken and germination poor. So you can see the symptom of anthracnose on the leaves also. Now management of anthracnose, seed treatment with carbendazim. Carbendazim is normally Bavastin 50 WP at the rate of 1 gram per kg of seed, or it can be done with Captan at the rate of 3 gram per kg seed. Either of these, Bavastin or carbendazim or Captan, can be used. And spray carbendazim 50 WP at the rate of 1 gram per liter, or Captan at the rate of 5 gram per liter, or Mancozeb 75 WP at the rate of 3 gram per liter. Means you can spray carbendazim or Mancozeb or Captan, one of these can be used. Next is soft rot. Attack begins when crop is 80 to 90 days old. Fungus first grows from soil and later slowly infects fallen leaves of jute. From there it infects stem base. Soft brown wet patch, it is wet patch, appears on stem base, skin peels off, exposed fiber layers turn rusty brown and plants will wilt. Stem becomes weak and often may break in high wind. Soft rot decreases if fallen leaves destroyed. Now you can see what are the symptoms of this. White cottony mycelium growth at stem. So you can clearly see the white growth of the fungus on the stem, particularly on the lower part of the stem. And you can see this later on, the breakage, the breaking of stem can happen and it can do severe damage to the crop. Now control measures for soft rot. Cultural control can be adopted. Deep plowing during summer season to expose sclerotia in soil to hot sun. Sclerotia are the propagules for survival of the fungus. Clean cultivation, remove leftover of previous crops. Means all kind of residues or stubbles of the previous crop should be removed from the field. Follow long-term crop rotation using non-host crops. Long-term rotation, for example, 3-year rotation, 4-year rotation can be followed. Suppose it is 4-year rotation, then every 4 years the jute crop should come only once in the same field. That is the meaning of 4-year rotation. Similarly, 3-year rotation means in every 3 years this particular jute crop will come only once in 3 years. Then it is 3 years rotation. So adopt green manuring with sunhemp or dhecha. Use farmyard manure or other organic manure like compost etc. during land preparation, but these manure should be mixed in soil at least 20-25 days before sowing of the crop. Soil solarization using polythene sheet increases inside soil temperature by 15 to 20 degrees centigrade, and use neem or mahua cake. Biological control with application of Trichoderma viridi in soil or through FIM. This Trichoderma viridi is a biocontrol agent. It is also a fungus, but it can eat other fungus. Now chemical control is spray copper oxychloride at the rate of 4 to 5 gram per liter directed towards basal region may contain spread. higher concentration of 2000 microgram per ml of mancozeb, complete inhibition of growth of pathogen was observed. Next is black band. Black band, dark colored black spots on stem with defoliation of plants. On rubbing spots with finger, black sooty powder adheres to the fingers which is not in stem root. So here you can touch the part and then there will be some sooty or black powder on your fingers. Stem may break at place of infection. So here on the right side you can see the picture. This is black band. It is not in continuity, it is in bands. If the infection goes deeper into the stem, then it can break down also. And there may be defoliation of the leaves as well. Now management of black band, seed treatment with carbendazim 50 WP at the rate of 1 gram per kg or mancozeb at the rate of 4 gram per kg seed. Or spraying can be done with copper oxychloride 50 WP at the rate of 4 gram per liter or carbendazim 50 WP at the rate of 1 gram per liter. These are quite effective fungicides, mancozeb and this copper oxychloride. Next is tip blight. As the name suggests, it starts from the tip of the plant or tip of the leaves. So blighting of newly emerged tender tissue at tip of young plants. Top soft folded leaves turn brown to black and water soaked, slowly rotten in high humidity. It is a minor disease, not a major disease of jute crop. Stem gall, as the name suggests, gall means kind of swelling. So swelling in stem can happen. First you can see on the right side in the picture. These are symptoms of stem gall. So green galls of diameter 4 to 10 centimeter on young stems, particularly at base where water comes in contact of the stem. Galls look lentil seed like outgrowth on stem. Growth slows as galls later turn rusty brown and plants wither and die. What can be done? Spray copper oxychloride 50 WP at the rate of 4 gram per liter. Next is hugly wilt or bacterial wilt. So drooping and wilting of leaves, stems soft, slimy fluid comes out on slight pressing. This is mostly symptom of bacterial diseases. Later plants defoliate, making naked stem with healthy roots. So you can see on the right side the infection of bacterial wilt. So you can see the plant is showing that there are no leaves. So defoliation has happened and finally the yield and quality of the product can be affected. Seed treatment with carbendazim 50 WP at the rate of 1 gram per kg and spray same fungicide at the rate of 1 gram per liter. Next is bacterial leaf spot caused by Xanthomonas campestris PV Nakatei corcori. Small dark green spots turned brown. So initially they may be dark green spot, but they will turn brown. You can see on the right side there are many on one leaf and they have turned now brown. And spots coalesced. Coalesced means later on they merge into each other to form necrotic area on leaves, turning yellow and dropped. Lesions on stem surrounded by yellow border causing girdling and death of plant above the infected part. Next is jute mosaic. As the name suggests, it is caused by virus. Known by names jute leaf mosaic, jute yellow mosaic, jute golden mosaic, and it is transmitted by whitefly. Small yellow irregular dots, also flakes, on leaf lamina in each early stage, which gradually increase and intermingled with green patches and give yellow mosaic. Plants appear pale, leaf size reduced, sometimes complete leaf may turn yellow. So these are symptoms of jute mosaic. And what can be done for the management? Rogging of infected plants. Rogging means just removing the plant and spraying of imidacloprid at the rate of 2 to 3 ml per 10 liter or thiomethoxam at the rate of 2.5 to 3 gram per liter to control whitefly. So to manage the jute mosaic, you need to control the vector. which is white fly, and it can be controlled by insecticide. So you can see the symptoms here. The yellowing of the leaves start from the picture 1, and then second it is increasing, you see, and size of the leaf is also small, and finally you get yellowing of the leaves. So this is jute mosaic. Next is root knot nematode. So white globular swellings on roots, you can see on the right side, globular swellings, kind of knots or swellings are there on the root. So these are symptoms of root knot nematode, and due to feeding of their larvae. So larvae feed here. Translocation of nutrients and water blocked. This is first important part, that these nematodes stop the uptake, translocation of water in the plant system. Therefore, plant growth will be affected. And plants yellow and stunted growth. Nematode facilitate entry of bacterial pathogen R. solanacearum and fungal pathogen M. fasciolina by injuring roots. So it is already discussed. Now, management of root knot nematode, carbafuran reduce nematode population. So carbafuran can be used at the rate of 30 kg per hectare, carbafuran 3 Cg. Organic amendments like cakes of karanj, mahua groundnut can be used, or even neem cake has been found useful. Removal of stubbles, weeding, thinning crop, etc. can be done. Now we go to the fiber quality of the jute. That is important topic. So quality of jute fiber between oletorious and capsularis varieties differ due to anatomical features of fiber cells and their orientation and genetically controlled. Fiber quality also influenced by soil, climate and crop nutrient management practices. So besides genetics, the management practices also affect the fiber quality, and definitely for farmers a good quality fiber will fetch more price. If quality of the fiber or economic product is poor, nobody will pay for it, and farmers will be in loss. So therefore quality becomes very, very important. So coarser and light body fiber is obtained from sandy soils, whereas fiber of superior quality is obtained from clay loam soils with silt. So therefore soil quality also affects the fiber quality. Many climatic parameters, many cultural practices and varieties may affect the quality, and also the rating. Rating was discussed elsewhere, which is also an important determinant of quality of the jute fiber. So besides soils, there are some other external factors, particularly the rating procedure. So single most important external factor controlling fiber quality is rating, and faulty rating marks positive contributions of variety, soil and climate. Under rating process, producers produce coarse fibers, while over rating gives dazed and weak fibers. So rating is a process of separation of fiber, or kind of extraction of fiber. Before extraction of fiber, rating is done. The bundles of jute are put in the water, particularly slowly moving water, and it is covered with some weight and also with some material like sesbania plants, or it can be banana leaves, or it can be some other plant, and then rating process start. And after that, manually or by machine, you can separate the fiber. Mostly by manual method, fiber is separated, and if there is over rating or under rating, then the quality of the fiber is affected. Similarly, the quality of water, if it contains more iron and more some other materials, then it can also deteriorate the quality of the fiber. Fiber quality is judged by its suitability for production of various types of yarn. Yarn is kind of threads and behavior in manufacturing process of end products. For assessing fiber quality of jute, 5 physical parameters are important, and these parameters are strength, fineness, how much fine fiber is, root content, sometimes basal portion of the roots of plant may come, defects in the fiber, and color. These are 5 important quality traits or parameters. Grading of fiber is done based on specific weightage allotted to these 5 parameters as per Bureau of Indian Standard. So, different weightages have been allotted to different parameters, and in totality the quality of fiber is assessed. So, these parameters are for example, strength. Strength, it is measured in gram per tex. This is the unit, tex is the unit, TEX, tex, and score is highest, which is 30. Defects, weight percent, it is 25. Root content, length percent, 20. Fineness is tex, 15. Color, whiteness index, color is measured by whiteness index and score is 10. So, overall you see it is 100 score given to 5 different parameters. Fiber strength is measured by fiber bundle strength tester. So, it is a machine, fiber bundle strength tester, breaking load of exactly 12.5 centimeter of fiber weighing 250 to 350 milligram, cut from middle portion of reed measured. If mass of fiber bundles are W1, W2, W3, W4 and W5 milligram and corresponding breaking loads are T1, T2, T3, T4 and T5 kg, then fiber tenacity or strength can be worked out by formula. You can see this formula. S is strength 125 into T1 plus T2 plus T3 plus T4 plus T5 bracket close divided by W1 plus W2, W3, W4, W4, all are added, or you can say it is 125 into summation T divided by summation W, gram per tex. So, this is the directly you can calculate the strength of the fiber. In present BIS, Bureau of Indian Standard Grading System, strength parameter gives 30 marks and has 4 subgroups as below. So, overall there are 30 marks, but there can be 4 groups 1, 3, 4, 5. 1 is excellent. When it is more than or equal to 25, it gets 30 marks, and for good it is less than 25 to 20, means more than 20, less than 20 to up to 15, and less than 15 is poor. You can see different scores are given. For poor score is 4, for average score is 13, for good 23, for excellent it is 30. Now, next is fiber fineness, one of most important component of fiber quality, which improves yarn properties and enhances spinability. It is usually expressed in tex, which is mass of a single filament of the length of 1 kilometer expressed in grams. So, this is how we can define the tex. In industry, it is expressed in denier, which is exactly 9 times the value in tex. Fiber fineness, being linear density of fiber, can be directly measured by gravimetric method, but it is time consuming. Airflow method is also available that can be utilized to determine fiber fineness in jute and mesta both. In present BIS grading system, fineness parameter gives 15 marks and has 3 subgroups. You can see on the next slide here value of tex instrumental, and you can see very fine. Very fine is less than or equal to 1.8 for white jute and less than and equal to 2 for tosa jute, and score is 15 full marks, and for fine it is more than 1.8 to 3 for white jute and more than 2 to 3 for tosa jute, score is 10, and more than 3, score is 5. Now, root content, hard barky region at lower end of reed. Reed is actually woody part of stem, end of reed is called root content. Instrumental method of measuring root content in present grading system is to cut hard barky bottom region of fiber reeds and measure length and express as percentage of length of reeds. In present BIS grading system, root content has 4 classes and given 10 marks. So, you can see there are 4 classes: excellent, good, average, poor. The range is for excellent it is less than or equal to 5, for good more than 5 but less than or equal to 8, for average more than 8 and less than or equal to 10, and for poor it is more than 10. Means higher the root content, poor is the quality. So in this case, if it is equal to or less than 5, then score is 20, full marks. For good it is 15, for average it is 8, and for poor it is 3. Next is defects. Factors which cause serious or partial damage to quality of fiber are known as defects. There may be certain defects in the fiber, there may be some breakage in the fiber, or there may be some lower strength at particular part. Defects usually detected in body of raw jute fiber, and defects are classified into two groups. One is major defects, other is minor defect. So major defects are dazed and overrated fiber. Fiber has been overrating has been done. 2 is center root, 3 is runner, and 4 is knots may be there, and entangled sticks and mosey fiber. These are actually considered as major defects. And minor defects include weak croppy end, gummy fiber, loose leaf, loose sticks and specks. So there are 5 minor defect and 6 major defect in the fiber. Now you can see subgroups with the score for defects of jute fiber. Quality, all these qualities have been hand and are based upon hand and eye. So you can see for excellent, defect should be minimum 0.5 and score is full 25. If it is 1, then 17 is the score. For 1.5, 9 is the score. For more than 1.5, 5 is the score. So more defect means poor quality, less defect means better quality or excellent quality. Color indicates degree of whiteness or brightness of fiber. It depends mostly on the rating situation and method of rating adopted, and also on the variety of jute. In laboratory, it is measured by the color and luster meter and equipment. Jute fiber in BIS grade system has been classified in 3 categories and given 10 marks. So you can see subgroup with the score for the color of the jute fiber, quality, hand and eye, value whiteness index through instrument. More than or equal to 65 is considered as good and it is given a score 10. And more than and equal to 45 to less than 65, it is 5 score, and for poor it is less than 45 and score is only 3. So then color description of jute in relation to scores. So white jute and tosa jute. So good is considered if it is more than or equal to 65, light creamy to white color may be in white jute, and in tosa jute light creamy to reddish white color for the good grade, and for average less than 65 to more than or equal to 45, brownish to reddish white with some light grey color in white jute and light grey to copper color in tosa jute, and lastly poor less than 45, grey to dark grey for white jute and grey to dark grey also for tosa jute. Dear students, I hope that you enjoyed this lecture. Thank you very much.